The purpose of this video is to introduce you to the fermentation test. We'll briefly speak about the components of the fermentation media and tube, and then discuss what you would be doing in lab. To begin, every fermentation test has a Durham tube within the larger screw cap culture tube. The Durham tube is inverted so that it is open on bottom and closed on top, and this will enable us to capture gases and determine whether or not the species we're testing is capable of producing carbon dioxide as a byproduct of fermentation. Every fermentation test has a fermentation broth, which is colored red. The cause of the red color is phenol red, a pH indicator, that has been added to determine if the species or strain we're testing is capable of producing acids as a byproduct of fermentation. A yellow result suggests that acids have been produced. Fermentation broth also includes a single sugar of interest to determine whether or not that species or strain is capable of fermenting that sugar. In this lab, we test for 3 different sugars: lactose, sucrose, and dextrose, which is also known as glucose. That means that you will add each species to 3 separate tubes to test each of the 3 sugars. As you've done in past labs up to this point in class, you would need to inoculate each of your fermentation tests with a species of interest. There are 4 species we test in lab: E. coli, A. faecalis, S. typhimurium, and S. aureus. The table in the bottom right is designed to show you that you would have a total of 15 fermentation tests, 3 for each species because of the 3 sugars we're testing, and then 3 tubes that are a negative control so that we have something to compare our results to. After inoculating each tube, we incubate for 24 hours. Now let's take a look at the results, beginning with our control.